Good morning, everyone. Today we'll be discussing subependymal giant cell astrocytoma, which comes under circumscribed astrocytic gliomas. Now, here I have two cases for you. So, case one is a 48-year-old lady who presented with raised ICT. Her coronal post-contrast sagittal and axial T1 weighted images show a well-defined heterogeneously enhancing T2 hyperintense mass in the region of foramen of Monro. She had multiple facial adenoma sebaceum on her face, these lesions, and also multiple subungual lesions. So we did intraoperative cytology of this tumor which we received in the lab. This case was operated by Dr. Virendra Singh Pawar from City Hospital, Kolhapur. So the smears were hypercellular and composed of tumor cells which were arranged in sweeping fascicles and sheets. The smears showed large plump tumor cells that have eccentrically placed vesicular nuclei. You can see these, all these are eccentrically placed with distinct nucleoli and abundant glassy cytoplasm. And we can see these hair-like projections from the ganglionic appearing cells. Again here, there are some fusiform cells, plump cells, polygonal cells. These polygonal cells, these resemble ganglion cells. Nuclear pleomorphism is present, multinucleated giant cells, spindle cells are here, and also strap-like cells are present. And the background is fibrillary and shows presence of lymphocytes here. No mitotic activity was. Histology. The tumor is composed of neoplastic cells arranged in sheets, nests, and fascicles, and the tumor shows combination of large plump cells along with small fusiform cells. The tumor has vascular stroma. You can see these are prominent blood vessels and also exhibited foci of calcification here, these two areas. The neoplastic cells have small round vesicular nuclei, and at places the chromatin appears clumped and moderate amount of eosinophilic cytoplasm. Whereas the large polygonal cells with ganglionic-like appearance, these have eccentric nuclei containing prominent nucleoli and abundant glassy cytoplasm. So we went ahead with immunohistochemistry. GFAP was expressed, S100 was expressed uniformly, IDH1 was negative, TTF1 positive, nuclear positivity was noted, and the Ki67 proliferation index was low. So we rendered a diagnosis of subependymal giant cell astrocytoma, CNS WHO grade 1. Case 2 is a 24-year-old lady who presented with one episode of seizures. Now, this case was operated by Dr. Harshad Chipre from Latur. The MRI revealed a well-encapsulated, predominantly solid tumor that measured 4.2 into 3.3 into 3.2 centimeters near the foramen of Monro, closely abutting the subependymal margin of the frontal horn of the lateral ventricle. The tumor appeared hypointense on T1 and heterogeneously hypointense on T2. Operative notes: left frontotemporal craniotomy and tumor excision by transcortical approach was done. We received 4 paraffin blocks for second opinion and immunohistochemistry. The microscopy revealed features of SEGA, that is sweeping fascicles of cells. The cells had astrocytic morphology, small round nuclei, and moderate amount of eosinophilic cytoplasm. The background is fibrillary. Now these are cells having ganglionic-like appearance. Yeah, these cells here which are polygonal in shape with eccentric nuclei and exhibiting prominent nucleoli. Here, these are the ganglionic-like cells, prominent nucleoli and glassy eosinophilic cytoplasm. These are the cells which have small round vesicular nuclei and eosinophilic, glassy eosinophilic cytoplasm. No mitotic activity was noted, and we went ahead with immunohistochemistry. GFAP S100 and TTF1 was positive. Synaptophysin was weakly and focally positive in ganglion-like cells. IDH1 was negative and the proliferation index was low. So this is TTF1 positivity here. So a diagnosis of SEGA, that is subependymal giant cell astrocytoma, CNS WHO grade 1, was given, and this tumor was at the foramen of Monro. So coming to discussion part, SEGA is a periventricular tumor composed of partly or large ganglion-like astrocytes and strongly associated with tuberous sclerosis. This definition is as per WHO CNS 5th edition. These correspond to CNS WHO grade 1 and majority are associated with TSC1 gene, that is tuberous sclerosis 1 gene, which is located on chromosome 9q34, and TSC2 gene located on chromosome 16p13 mutations. Localization: subependymal region of the lateral ventricle near the foramen of Monro with extension into the lateral ventricle. These are rarely found around the third ventricle. Rare extraventricular masses have been reported either sporadically or in the patients with tuberous sclerosis. These can be bilateral. Retina is the rare possible site. Subependymal nodules, that is SENS, is found in more than 90% of patients of tuberous sclerosis. SEGA may arise from SENS almost always near the foramen of Monro, and the size varies. These are slow-growing tumors often present when 2 to 3 centimeters in size and cause hydrocephalus. These typically occur in the first 2 decades of life, showing no gender preference, and represent 1 to 2% of pediatric brain tumors, often occur with tuberous sclerosis. SEGA is the most commonest CNS tumor in patients with asymptomatic tumors are identified during regular screening for TSC. Symptoms of raised ICT are present. Acute manifestation is due to massive spontaneous hemorrhage, and they may also present with imaging findings. SEGA appears as an enlarging, enhancing foramen of Monro mass in patients with tuberous sclerosis complex, typically more than 1 centimeters in size. On MR, these are T1 hypointense to isointense to the gray matter, and foci of calcification appears as hyperintense to hypointense. Whereas on T2 weighted images, SEGA appear as heterogeneous iso to hyperintense, whereas the calcium foci appear hypointense. Hydrocephalus may be present. Other imaging findings such as cortical tubers or subependymal nodules may be found. Uh, Let us know little about SENS. These are typically 5 to 10 mm in size. SEGAs usually grow slowly while SENS remain normal. Morphologically, these are well marginated, often lobulated, and have front-like margins. Differential diagnosis on imaging. Choroid plexus papilloma is the differential diagnosis. It commonly occurs in the atrium of the lateral ventricle, and foramen of the Monro is a rare site. Next is astrocytoma. Origin is septum pellucidum fornicis and medial basal ganglia. Next is the germinoma. It hugs the midline, often arises near the third ventricle, and it shows early CSF spread. Subependymoma. It is a tumor of the middle-aged or the elderly adults, and it is more common in the inferior fourth or the frontal horn of the lateral ventricle. Next is the central neurocytoma. These are well-defined, variably vascularized, lobulated masses. Body of the lateral ventricle is more commonly affected than the foramen of Monro or septum pellucidum, and it is commonly seen in the young adults. Diagnostic molecular pathology is usually not needed for diagnosis of SEGA. DNA methylation profiling and analysis of TSC1 and TSC2 alterations may help establish diagnosis. SEGA is a solitary, slow-growing benign tumor. Patients with gross total resection have favorable prognosis. In individuals with tuberous sclerosis, surveillance by MRI every 1 to 3 years until the age of 25 years is recommended. mTOR inhibitors such as Everolimus show significant reduction in the tumor size, and rarely there may be massive spontaneous hemorrhage. If you like this case, please share this with your colleagues and subscribe my channel. Thank you very much.